on this video if something is easier or harder. So identify the parts of the nucleotide. There are 3 parts to the nucleotide. We have our phosphate group, which is usually a circle, and that is made of phosphorus and 4 oxygen. And then we have our 5 carbon sugar, which is our pentagon. And the elements in that are C, H, and O, because remember, sugar is C6H12O6. And then this will, This will be the components of our backbone. And then finally, we have our N base, which is either a purine or a pyrimidine, and it's A, T, C, and G. So the macromolecule of DNA is going to be nucleic acid. And then the monomer is going to be a nucleotide. And remember, our nucleotide has 3 parts, and our nucleotide is made up of covalent bonds. So, covalent bonds bond the nucleotide together. Okay, which elements make up DNA? So we talked about it on question 1, but we have phosphorus, oxygen for our phosphate, and then we have CHO in our 5 carbon sugar. So we have hydrogen, we have carbon, and then we have nitrogen in our nitrogenous base. So if you wanted to write that in our little acronym, it's carbon, hydrogen, oxygen, nitrogen, and phosphorus. Chomp. Describe the characteristics of an H bond and identify where it is in the DNA molecule. So H bonds are those circle dotted bonds. They are weak, and they are on the rungs of our ladder. So they bond complementary base pairs together. Describe the characteristics of a covalent bond. So covalent bonds, remember, they're going to be solid lines. They're strong. And then they are located in the nucleotides. In the nucleotides, so it's really any solid line you see throughout, but they're in nucleotides, and they bond the backbone together. Because remember, backbones are super strong. Our backbone's really strong. What is the central dogma of biology? So we go DNA to RNA to protein. So essentially what we're saying is that DNA makes proteins. So DNA makes proteins. So this is indirectly because RNA is part of it, but we have that nucleic acid to protein. And the way that this takes place is replication, which is DNA to DNA. So DNA to RNA is called transcription. So we're transcribing the DNA to an RNA molecule, and then we're translating that nucleic acid blueprint to protein via translation. So replication to transcription, and we'll learn about transcription and translation next unit. So transcription and then translation. And this is all how we make proteins from DNA. So where is DNA located in eukaryotes? Nucleus, so that's kind of the brain of the cell, right? What are chromosomes and how do they relate to DNA? So chromosomes are made of DNA. So we put all of our DNA together, condense it up, and then we can make a chromosome. So chromosomes are larger than DNA if you were trying to put it into context. Who discovered the structure of DNA? So we'll start off with number 2. So in 1950, Chargaff talked about species have the same number of A and T and C and G. So same number of nucleotides. And that makes a species a species. So Chargaff looked at species and he looked at their base pairs and how they match. And then Watson and Crick, 3 years later, discovered that they used Chargaff's principle and then discovered the double helix anti-parallel structure. Parallel structure. That's actually the wrong way to write parallel. If you're in math, you write it like this, right? Parallel structure. Okay, identify the purines and the pyrimidines. So A and G, purine. C and T, pyrimidine. And remember, this will be double halo, and this is single halo. Label the 5' and 3' ends of the carbon sugars. So remember, we have 5. Sorry, hold on. Okay, so the 5' and 3', we have 1, 2, 3, 4, 5. So 5', 3'. So remember, 5' is where we're pointing up and 3' is down. Describe anti-parallel. So essentially what that's saying is that we have one strand that our pentagons are facing one way. And then on our other strand, our pentagons are facing the opposite way. So we have 2 parallel pieces of DNA. These are parallel, right? But they are opposites, right? Because we look at the way the pentagons are facing. So they're anti-parallel. So they're parallel, but actually you just flip them. So they're anti-parallel. And this helps us because we can label this 5' and then 3', 5', 3'. So the directionality has to do a lot with the anti-parallel. Also those bases, remember our A bonds with our T. So we know due to the anti-parallel nature, we can tell which base will bond with which base. So replication, what, where, when, why. So DNA to DNA is what? Why all cells need DNA. Where nucleus inside a eukaryote, and when S phase, because we are synthesizing DNA. Identify the leading strand and the lagging strand in relation to the fork. So the leading strand will have 5' closest to the fork, and then the lagging strand will have 3' closest to the fork. So it's just opposite, closest to fork. Compare the directionality of template strands and newly synthesized strands. So if we have a template strand that is 5' to 3', the newly synthesized strand will be 3' to 5'. In which direction do we build DNA? So we're always building it from 5' to 3'. Why does the lagging strand create Okazaki fragments? So the lagging strand is creating Okazaki fragments because we are doing the jumps. Because remember, we're creating DNA away from fork. So we're running out of room. Thus, our lagging strand is going to have to jump. So essentially what's happening is we are replicating this way. So we run out of room, so we're replicating, boom, and then now we have to jump forward, and then we replicate, and then we boom, jump forward, replicate. So it's because we are growing in the opposite direction the DNA is growing. So, the DNA is being unzipped, so that's why we have to do our jumps, and that gives us these fragments, and then who is going to glue them together? Ligase. And then for the opposite reason, the leading strand creates a continuous newly synthesized strand because it is replicating towards the fork and it's expanding. What is an enzyme? So an enzyme is a protein that speeds up / helps chemical reactions. And remember, chemical reactions are the building up and the breaking down of molecules. So we build up molecules, break them down, and enzymes help us do that. So enzymes can be reused. Remember, they often end in ASE. Macromolecule, just said that, proteins. And then discuss the purpose of the enzymes. So helicase, unzip. Remember, there's just going to be one. There's just one. Ligase, glue. If we're why did I put glucose? That's not it. So ligase will glue, and then that means that ligase only on the lagging strand, and there can be multiple ligases. There will be in your diagrams. Can be multiple. DNA polymerase is the builder. It's going to be on both strands because we're just building. And then primase is the starting point. So, if you're still with me, listen to this point. So when we're thinking about our leading strand, we have a continuous strand like this. So we have one starter, right? Okay, well that's our leading strand. Our lagging strand, if we're building in the opposite direction, so we're going this way, oops, we ran out. So we have to start again, boop, we ran out. Start again, boop, ran out. Start again, boop, ran out. Start again. So how many primases do we have here? Well, we have 5 primases because of those Okazaki fragments. So this was just a quick little review, but I highly recommend listening to this whole thing because I talk through a lot more than what I write on this. So I hope that this helped. And good luck on the exam tomorrow. I think that y'all will do great.